So we have a range of Kaha Sciences RAT telemeters available. We have telemeters that can record biopotential signals, pressure signals, sympathetic nerve activity and tissue oxygen. All of our telemeters are shipped sterile and ready to go. All of the telemeters are encased in a biocompatible polymer, which makes them robust to implantation and suitable for the implantation environment. Those cases all have some suture tabs, and that enables you to secure the telemeter within the abdomen of the rat for optimal charging performance. All of the telemeters are capable of receiving wireless power from the SmartPad, and they also all contain a backup battery, which, depending on the telemeter model, will give you 4 to 6 hours of recording away from the SmartPad. All of the telemeters are designed to be reused, so that at the end of your experiment, you can explant them, clean them up, re-sterilise them, and use them again for your next experiment. There's no refurbishment or need to send them back to the manufacturer. This is the TR50B single biopotential RAT telemeter. This can be used to record a single biopotential signal. So it records the potential difference between the 2 electrode leads. If you were to record ECG, you could put them across the chest. EMG, you could choose a muscle to place the electrodes across, or EEG to record signals from the brain. The telemeter has suture tabs to hold it in place in the abdomen, and then the leads are tunneled to your recording site of interest. At the end of your experiment, you can explant the telemeter, trim the leads back slightly, re-sterilise your telemeter, and perform your next recording. This is the TR50BB dual biopotential RAT telemeter. It contains 2 pairs of biopotential leads, the red and the black, and the green and the yellow. And these leads are placed wherever you want to record a biopotential signal. For example, you may choose to place the red and the black leads into the brain to measure an EEG signal, and attach the green and yellow leads to the nuchal muscle at the back of the neck to measure EMG. And in this way you could measure EEG signals from the rat and use the EMG signals to detect between the wake and sleep cycles. The telemeter has suture tabs to hold it in place in the abdomen, and then the leads are tunneled to wherever you're making your recording. At the end of your experiment, you simply remove the telemeter, trim the leads back to expose a new piece of electrode wire, and re-sterilise and reuse your telemeter. This is the TRM54PP dual pressure RAT telemeter. We also have available a TRM54P single pressure telemeter, which only has 1 pressure catheter. Our pressure catheters come in 3 options of sizes. This is the 9 centimetre pressure catheter, which is the standard size. We also have a 15 centimetre catheter and a 25 centimetre catheter available. The 9 centimetre catheter is ideal for placing in the abdominal aorta of the rat for measurement of arterial pressure. And you can also place it up through the diaphragm into the left ventricle of the rat to record left ventricular pressure. The 15 centimetre catheter is a good alternative if you're wanting to record from the femoral artery, or if you're wanting to record something like venous pressure as well as arterial pressure. The 25 centimetre option is perfect for use for recording intracranial pressure, as it needs to be tunneled from the abdomen of the animal up to the skull to record the intracranial pressure. All the Kaha Sciences pressure telemeters include a solid-state high-fidelity pressure sensor. This Miller microtip pressure sensor is specifically designed to be fully implantable within the rat for chronic experiments. The solid-state pressure sensor is ideal for recording something like intracranial pressure, because it's such a low pressure signal that any effect of gravity on a fluid-filled catheter will change your reading of your intracranial pressure, whereas you don't get that with the solid-state pressure sensor. It's also ideal for recording signals such as left ventricular pressure, because it has a high fidelity signal and a high to PDT values can be picked up accurately. It's very easy to clean and reuse the solid-state pressure sensors. There's no need to re-gel the tip or to remove bubbles or clots. You just simply clean the sensor, re-sterilise it and reuse it, and you're ready to go. This is the TRM54PB pressure biopotential telemeter. And this is useful for recording for a pressure signal using the pressure sensor and a biopotential signal using the biopotential leads. And a good example of this would be to record abdominal aortic pressure or arterial pressure from the abdominal aorta and ECG from the chest of the animal. So the telemeter body would be placed in the abdomen and kept in place with the suture tabs, and then the leads and catheter tunnel to wherever your recording site is. This is the TRM56SP pressure sympathetic nerve activity telemeter. It includes a solid state pressure sensor catheter for measuring arterial pressure and a high gain amplifier for measuring sympathetic nerve activity. So you would place the multi-stranded Kuna wire electrodes around your sympathetic nerve, and then the coiled electrode gets placed nearby on the muscle for measuring the earth reference point. An example for this would be to record renal sympathetic nerve activity and arterial pressure and look at the changes to those as hypertension develops. This is the TR57Y tissue oxygen telemeter. It essentially acts as a potentiostat, so it applies a fixed voltage of 650 millivolts to the electrodes and measures the current response. And that current is directly proportional to the amount of oxygen in the tissue. The blue electrode has a carbon paste electrode attached, and that is placed wherever you want to record your tissue oxygen. So that might be in the kidney or in the brain, for example. The platinum silver electrodes on the red and the black electrode leads, they act as the reference and the auxiliary leads to complete the circuit. The telemeter is placed in the body of the rat in the abdomen with the suture tabs to keep it in place, and then the leads are tunneled to wherever you're wanting to record your signal from. The telemeters come with a set of electrodes attached, but between each use you just simply trim the electrodes off and reattach a new set of electrodes, re-sterilise and perform further recordings. The latest telemeter in the Kaha Sciences rat telemeter range is the Optogenetics telemeter. This is the TR58AB Optogenetics biopotential rat telemeter. It includes a single pair of biopotential recording electrodes and an optical fibre to deliver blue light at 460 nanometers wavelength. This can be used to activate the opsins of channel rhodopsin in neurons which have been transfected with a virus. You may want to use the optical fibre implanted in the brain to activate an area of interest and measure the biopotential leads to record the EEG from the brain in response to the chronic optogenetic stimulation.